Welcome to the fascinating world of genome editing. Today we're diving into CRISPR-Cas, a groundbreaking technology that's transforming biology. CRISPR, short for clustered regularly interspaced short palindromic repeats, is a tool that allows scientists to make precise changes to DNA. Imagine being able to edit the genetic code as easily as correcting a typo in a document. This technology is opening doors in medicine, agriculture, and beyond. In this video, we'll explore how CRISPR works, its origins, and the incredible possibilities it offers. Let's begin our journey by understanding what makes CRISPR so revolutionary. Story of CRISPR begins in the late 1980s, when scientists noticed unusual repeating sequences in the DNA of bacteria. These sequences, later named CRISPR, puzzled researchers for years. It wasn't until the early 2000s that scientists realized these repeats were part of a bacterial immune system, helping bacteria defend against viruses. This discovery was a turning point, revealing that bacteria could remember and cut up viral DNA. Understanding this natural defense mechanism laid the foundation for developing CRISPR as a gene editing tool. Cas9 is a protein that acts like molecular scissors. In the CRISPR system, Cas9 is guided by a piece of RNA to a specific spot in the DNA. Once it finds its target, Cas9 cuts the DNA at that exact location. This precise cutting ability is what makes CRISPR-Cas9 such a powerful tool for genome editing. Scientists can program the system to target almost any gene, making it incredibly versatile for research and therapeutic application. CRISPR-Cas9 system works by introducing a guide RNA that matches the DNA sequence scientists want to edit. This guide RNA leads Cas9 to the exact spot in the genome. Once there, Cas9 makes a cut in the DNA. The cell then tries to repair this break, and scientists can use this opportunity to add, remove, or change genetic material. This process is fast, efficient, and can be used in many different organisms, from bacteria to plants to animals. CRISPR's origins lie in the natural world. Bacteria use CRISPR as a defense system against viruses called bacteriophages. When a virus attacks, bacteria capture snippets of the viral DNA and store them in their own genome. If the same virus attacks again, the bacteria use these stored sequences to recognize and destroy the invader. This elegant system inspired scientists to adapt CRISPR for genome editing in other organisms. Guide RNA is a crucial component of the CRISPR-Cas9 system. It's a short piece of RNA that matches the target DNA sequence. The guide RNA acts like a GPS, directing Cas9 to the precise location in the genome where the cut should be made. By designing different guide RNAs, scientists can target almost any gene, making the system highly customizable and powerful for research. One of the most exciting aspects of CRISPR-Cas9 is its precision. Unlike older gene editing methods, CRISPR can target specific genes with minimal off-target effects. This accuracy reduces the risk of unintended changes and makes the technology safer for use in medicine and agriculture. Scientists are continually improving the system to make it even more precise and reliable. CRISPR-Cas9 is revolutionizing medicine. Researchers are using it to develop new treatments for genetic diseases such as sickle cell anemia and cystic fibrosis. By correcting faulty genes, CRISPR offers hope for curing conditions that were once thought to be untreatable. Clinical trials are underway, and the results so far are promising. The potential to eliminate inherited diseases is one of the most exciting prospects of genome editing. Beyond medicine, CRISPR is making waves in agriculture. Scientists are using genome editing to create crops that are more resistant to pests, diseases, and environmental stress. This means higher yields and less reliance on chemical pesticides. CRISPR can also be used to improve the nutritional value of food, helping to address global challenges like hunger and malnutrition. CRISPR is also being used to edit the genomes of animals. This has applications in both agriculture and medicine. For example, scientists can create livestock that are more disease-resistant or produce healthier meat. In medical research, animal models with specific genetic changes help scientists study diseases and develop new treatments more effectively. Gene drives are a powerful application of CRISPR technology. They allow scientists to spread a specific genetic trait through a population much faster than normal inheritance would allow. This could be used to control pests or disease-carrying insects, like mosquitoes that transmit malaria. However, gene drives also raise important ethical and ecological questions that must be carefully considered. With great power comes great responsibility. The ability to edit genomes raises significant ethical questions. Should we use CRISPR to alter human embryos? What are the risks of unintended consequences? Scientists, ethicists, and policymakers are working together to develop guidelines and regulations to ensure that genome editing is used safely and responsibly. While CRISPR offers incredible promise, it's not without risks. Off-target effects where unintended parts of the genome are edited can cause problems. There's also the possibility of unintended ecological impacts, especially when editing wild populations. Ongoing research aims to minimize these risks and ensure the technology is as safe as possible. Scientists are constantly working to improve the accuracy of CRISPR-Cas9. New versions of Cas proteins such as Cas12 and Cas13 offer even greater precision and flexibility. These advances help reduce off-target effects and expand the range of possible genome edits. The field is evolving rapidly, with new discoveries being made every year. CRISPR isn't just for editing genes, it's also being used to develop new diagnostic tools. By harnessing the ability of Cas proteins to recognize specific DNA or RNA sequences, scientists can create rapid, accurate tests for diseases like COVID-19. These CRISPR-based diagnostics are fast, affordable, and can be used in a variety of settings. CRISPR is opening up new possibilities for treating a wide range of diseases. From genetic disorders to cancer, researchers are exploring how genome editing can correct faulty genes or disable harmful ones. Some therapies are already in clinical trials and the results are promising. The hope is that CRISPR-based treatments will one day become routine in medicine. Many rare genetic diseases have no cure, but CRISPR offers new hope. By targeting and correcting the specific mutations that cause these conditions, scientists can potentially cure diseases that affect only a small number of people. This personalized approach to medicine could transform the lives of patients and families affected by rare disorders. Gene therapy is one of the most exciting areas of CRISPR research. By delivering the CRISPR-Cas9 system directly into patient cells, scientists can repair or replace faulty genes. Early successes in treating blood disorders and inherited blindness are paving the way for more breakthroughs in the future. Editing the genomes of embryos is one of the most controversial uses of CRISPR. While it could prevent inherited diseases, it also raises ethical and safety concerns. Most countries have strict regulations or bans on editing human embryos, but research continues in some areas to better understand the risks and benefits. The idea of using CRISPR to create designer babies' children with selected traits has sparked intense debate. While the technology could eliminate genetic diseases, it also raises concerns about inequality, consent, and the definition of what it means to be human. Society must carefully consider the implications before moving forward. Cancer research is benefiting greatly from CRISPR technology. Scientists can use genome editing to study how cancer develops and to identify new drug targets. In some cases, CRISPR is being used to engineer immune cells that can better recognize and attack cancer cells, offering new hope for patients. CRISPR is also being used to fight infectious diseases. By editing the genomes of viruses or the cells they infect, scientists can develop new treatments and even potential cures. This approach is being explored for diseases like HIV, hepatitis, and more, offering new strategies for global health. CRISPR isn't just for health and agriculture, it's also being used to address environmental challenges. Scientists are exploring ways to use genome editing to clean up pollution, protect endangered species, and even combat climate change. These innovative applications could have a significant impact on the planet. Public understanding and acceptance of CRISPR technology are crucial for its success. While many people are excited about the possibilities, others have concerns about safety, ethics, and unintended consequences. Open communication and education are key to building trust and ensuring responsible use of genome editing. As CRISPR technology advances, governments and international organizations are working to develop regulations and oversight mechanisms. These rules are designed to ensure that genome editing is used safely and ethically, balancing innovation with public safety and societal values. CRISPR regularly makes headlines as new breakthroughs and controversies emerge. From successful treatments to ethical debates, the technology is at the center of scientific and public attention. Staying informed about the latest developments is important for understanding the impact of genome editing on society. The development of CRISPR technology has led to intense competition over patents and intellectual property. Universities, companies, and inventors are all vying for control over key aspects of the technology. These legal battles can influence who has access to CRISPR and how it's used in research and industry. As CRISPR becomes more widely used, education and training are essential. Scientists, doctors, and technicians need to understand how the technology works and how to use it safely. Universities and research institutions are developing new courses and programs to meet this growing demand. Biotechnology companies are rapidly adopting CRISPR for a wide range of applications. From developing new medicines to engineering microbes for industrial processes, the technology is driving innovation across the biotech sector. This growth is creating new opportunities and challenges for the industry. The future of genome editing is bright. As CRISPR technology continues to advance, we can expect even more precise, efficient, and safe methods for editing DNA. New discoveries and applications are on the horizon, promising to reshape medicine, agriculture, and many other fields. Synthetic biology is another area where CRISPR is making a big impact. By combining genome editing with synthetic biology techniques, scientists can design and build new biological systems from scratch. This could lead to the creation of custom organisms with useful properties for industry, medicine, and the environment. CRISPR is paving the way for personalized medicine, where treatments are tailored to an individual's genetic makeup. By understanding and editing specific genes, doctors can develop therapies that are more effective and have fewer side effects. This approach has the potential to revolutionize healthcare. Genome editing with CRISPR has the potential to influence evolution itself. By making targeted changes to the DNA of plants, animals, and even humans, we can shape the traits of future generations. This power comes with great responsibility and requires careful consideration of the long-term consequences. CRISPR research is a global effort, with scientists from around the world collaborating to advance the technology. International partnerships are essential for sharing knowledge, setting standards, and addressing challenges that cross borders. Working together, the scientific community can maximize the benefits of genome editing. Genome editing generates vast amounts of data. Analyzing this information is crucial for understanding the effects of CRISPR edits and ensuring safety. Advances in data analysis and bioinformatics are helping scientists make sense of complex genetic information and improve the accuracy of genome editing. Engaging the public in discussions about CRISPR is vital. By involving people in conversations about the benefits, risks, and ethical considerations, we can ensure that genome editing reflects society's values and priorities. Public engagement helps build trust and supports responsible innovation. CRISPR is becoming a topic in classrooms around the world. Educators are introducing students to the basics of genome editing, inspiring the next generation of scientists and innovators. Hands-on activities and interactive lessons help make this complex topic accessible and exciting. Open science initiatives are making CRISPR research more accessible. By sharing data, methods, and results openly, scientists can accelerate progress and ensure that the benefits of genome editing are widely shared. Collaboration and transparency are key to the future of CRISPR. Bioethics plays a central role in guiding the development and use of CRISPR technology. Ethical frameworks help scientists and policymakers navigate complex questions about safety, consent, and the impact on future generations. Ongoing dialogue is essential for responsible innovation. Legal systems are adapting to the challenges posed by genome editing. Laws and regulations are being updated to address issues like intellectual property, safety, and access to CRISPR technology. Legal experts are working alongside scientists to ensure that the law keeps pace with scientific advances. The economic impact of CRISPR is significant. The technology is creating new industries, jobs, and investment opportunities. From biotech startups to large pharmaceutical companies, CRISPR is driving economic growth and innovation around the world. Despite its promise, CRISPR faces many challenges. Technical hurdles, ethical debates, and regulatory uncertainties must all be addressed. Continued research, collaboration, and public engagement are essential for overcoming these obstacles and realizing the full potential of genome editing. Genome editing with CRISPR has the potential to shape the future of humanity. From curing diseases to enhancing crops and protecting the environment, the possibilities are vast. As we move forward, it's important to balance innovation with responsibility, ensuring that the benefits of CRISPR are shared by all. In summary, CRISPR-Cas technology is revolutionizing genome editing. Its precision, versatility, and wide range of applications make it one of the most important scientific advances of our time. As we continue to explore its potential, we must also consider the ethical, legal, and social implications. The future of genome editing is in our hands. Thank you for joining us on this journey through the world of CRISPR-Cas genome editing. The technology is still young and its story is just beginning. With continued research, collaboration, and thoughtful discussion, CRISPR has the potential to change our world for the better. Stay curious and keep exploring the possibilities of science.